A very warm good evening to one and all. I welcome you all to the seventh day of 1 week online faculty development program on application of next generation sequencing technologies in microbial genomics, organized by Center for Ocean Research, Satyabama Institute of Science and Technology in association with Ministry of Earth Sciences, Earth Science and Technology Cell. I welcome the key speaker of today's evening, Ms. Maithili Verma. Ms. Maithili has research experience working in various projects in the areas of phylogenetics, industrial biotechnology, clinical research, molecular modeling, etc. She has expertise in gene technology, protein technology, and bioinformatics, and has also presented seminars in varied domains. She is also having industrial exposure working in reputed companies as intern or trainee in the fields of bioinformatics, bioengineering, and next generation sequencing technologies. I request Ms. Maithili Verma to deliver her talk. Over to you, ma'am.
Thank you for that warm welcome. And I will be sharing my screen now. I would also like to thank Mr. Thiru for giving me this opportunity to speak on this platform. And I will be sharing my screen right now. Can it be seen?
Yes, ma'am. Please proceed.
Okay, I will be talking about next generation sequencing, the history, tools, and omics approach. So this is the outline of my talk. I will be talking about the introduction to NGS, history of NGS, first generation sequencing, second generation sequencing, then third generation sequencing or single molecule sequencing, then bioinformatics, that's data analysis and storage, and also the omics technologies. So coming to an introduction to DNA sequencing. What is DNA sequencing? The process of determining the sequence of nucleotides, that's adenine, thymine, cytosine, and guanine, in a piece of DNA is called DNA sequencing. Next, coming to the history of DNA sequencing. The history of DNA sequencing begins with the discovery of the DNA structure in the year 1953. And in the year 1977, Frederick Sanger postulated the first method for the sequencing of the DNA, named the chain termination method. In the same year, Maxim and Walter Gilbert sequenced the genome of the bacteriophage X174 using the chemical degradation method. Both these methods were tedious and time-consuming because of the lack of automation. So later on, in the year 1987, Applied Biosystems had developed a fully automated machine-controlled DNA sequencing method. And then the era of the 2000s became the golden period for the sequencing platforms. So in the year 1996, Applied Biosystems had developed another innovative sequencing platform, which was known as capillary DNA sequencing. After that, the Human Genome Project was completed by using a combination of all these methods in the year 2003. In the year 2005, the fast, accurate, reliable, and highly efficient next generation sequencing technology was postulated by the Solexa Illumina. So coming to an introduction to NGS. What is NGS? Next generation sequencing refers to the deep high throughput in parallel DNA technologies that's developed a few decades after the Sanger sequencing method was first emerged in 1977. What are the advantages of NGS? It is a high throughput DNA sequencing technique. It employs both micro and nanotechnologies, and only a small amount of the sample is necessary for sequencing. The cost of the reagents are also low, and the entire technique requires less time than the older sequencing techniques. It involves massive parallel sequencing, and the sequences it sequences thousands of sequences at once, and the amount of data that's produced is enormous. So coming to the first generation of sequencing. This first generation of sequencing is called Sanger sequencing, and it was developed in the year 1977 by Frederick Sanger. This technique was also known as the Sanger sequencing technique or the dideoxynucleotide chain termination technique. So this technique was used for commercial sequencing applications as well as in laboratories because of its high efficiency and low radioactivity. Now coming to the differences between, so the Maxim and Gilbert sequencing technique involves the nucleotide specific partial chemical modification and then DNA cleavage, whereas Sanger sequencing is the process of incorporation of chain terminating dideoxynucleotides by the DNA polymerase during DNA replication. The Maxim and Gilbert sequencing method was developed by Alan Maxim and Walter Gilbert in the year 1977 to 1980, whereas the Sanger sequencing method was developed by Frederick Sanger in the year 1977. And the Maxim and Gilbert sequencing technique is a chemical method of sequencing, whereas the Sanger sequencing technique is a chain termination method. So also the Maxim Gilbert sequencing technique is highly sensitive and highly specific, whereas the Sanger sequencing technique is less sensitive and less specific. So coming to what is a dideoxynucleotide or DDNTP. A dideoxynucleotide or DDNTP is an artificial molecule that lacks a hydroxyl group that's at the 3' carbon of the ribose sugar. This hydroxyl group is needed for the attachment of another deoxynucleotide so that the chain grows. And this is why the dideoxynucleotide or DDNTPs are known as chain terminators, because they stop the chain from growing. Then coming to the steps in Sanger sequencing. The first step in Sanger sequencing is to denature and clone. So the first step is to fragment the DNA by using heat and then clone the fragments into vectors. And the next step is primer attachment. So the second step is to anneal this synthetic oligonucleotide to a target sequence. And this oligonucleotide then acts as a binding site for the primer, and it also provides a 3' hydroxyl group, which is necessary for DNA synthesis. Then coming to the third step involves the addition of DNTPs and DDNTPs. So we have 4 different reaction vials which are used, and each with 4 standard DNTPs and the DNA polymerases. So the difference between these vials is the type of the DDNTPs that are used. So each vial will have 1 DDNTP per 100 DNTPs. So in general, we could say this vial will contain a DATP, DCTP, DGTP, DTTP, the DNA polymerase, the template DNA, and a primer. But the only difference is the DDNTPs that are present in each of the vials. So after that, after DNA synthesis occurs, each reaction vial will have a unique set of single-stranded DNA molecules of varying lengths, and all the DNA molecules will have the same primer sequence, however, and the DNA molecules, it'll have the same primer sequence at its 5' end. Then the resulting DNA fragments are denatured by heat, and these base paired loops of single-stranded DNA, because these base paired loops will have, it'll be difficult to resolve them while running a gel. So additionally, you can use formamide to prevent the base pair loops forming. So the next step is gel electrophoresis. And these sequences are separated based on their size to determine the sequence, or the fragments are they're separated based on their size to determine the sequence. and radiography is used. Here, the DDNTPs are radioactively or fluorescently labeled beforehand for automated sequencing machines. Then the DNA strands are separated using gel electrophoresis, and it is read from top to bottom, that is from the 3' to 5' end to obtain the sequence. So each DDNTP will be fluorescently labeled, and this causes each of the 4 DDNTPs to emit light at different wavelengths. So here in capillary electrophoresis, uh, this is carried out in a single lane to capture the nucleotide sequence. And you can see here that the primer sequence is the same, and but even the sequences, the target sequence is the same, but they are cut at different positions throughout this, and then you can see that it is read out through capillary electrophoresis. So coming to the second generation of sequencing, it's also called next generation of sequencing. So NGS generates many millions of short nucleotide short reads in parallel in much shorter time than the Sanger sequencing method. But the basic characteristics of NGS is that the random fragmented DNA or the cDNA, which is reverse transcribed from the RNA, is performed without the need for cloning into a foreign host cell. Instead, we use linker adapter sequences that are ligated to the ends of the fragmented DNA or the cDNA to construct the template libraries. And also nucleotide incorporation. This nucleotide incorporation is monitored by luminescence detection or by changes in electric current during the sequencing procedure. So now coming to the different types of technologies in NGS. One is Roche 454 pyrosequencing by the 454 Life Sciences, which was in the year 2005. Then Illumina or Solexa sequencing, that's mainly HiSeq and MiSeq sequencing. It was commercialized in the year 2007, and it was purchased by the Solexa Genome Analyzer in the year 2006. Then there's sequencing by oligonucleotide ligation and detection called SOLID. And there's nanoball sequencing by BGI retrovelocity. And there's also ion torrent by the inventors of 454 pyrosequencing. So I will be talking about a few of these technologies in more detail in the coming slides. So coming to Roche 454 sequencing, pyrosequencing. So pyrosequencing relies on light detection based on a chain reaction when pyrophosphate is released during repeated nucleotide incorporation, and that is when incorporation into the newly synthesized DNA chain. So the DNA fragments are attached to the beads, and the fragments are then amplified by PCR. Each bead is covered in many PCR copies of a single sequence, and then it is placed in a single well of a microfabricated microarray. Then the next step is the slide is flooded with one of the 4 nucleotides, and the addition of each nucleotide then releases a light signal, and these locations of the signals are detected and used to determine which beads the nucleotides are added to. The next step is this nucleotide mix is washed away, and the next nucleotide mix is then added, and the process is repeated, cycling through the 4 nucleotides. So in general, the nucleotide is added, and then it releases a light signal, and then that nucleotide is washed away, and the second nucleotide is then added. So this then generates a graph, which is called the read flowgram for each sequence read, showing a signal density for each nucleotide wash, and a sequence can then be determined computationally from the signal density in each wash. So coming to Illumina or Solexa sequencing. Illumina purchased the Solexa genome analyzer in the year 2006, and it commercialized it in the year 2007. It is the most successful sequencing system with 70% dominance of the market. That's mainly the HiSeq and MiSeq systems. And it uses the technology of sequencing by synthesis. That is, it uses removable, and it also uses removable fluorescently labeled chain terminating nucleotides. And this template DNA is amplified by the process of bridge amplification and or cluster generation, and this results in miniature colonies, which are called polonies. Also, the lengths are much shorter, approximately like 100 base pairs, and it is much cheaper than the other sequencing system. So coming to the steps in Illumina sequencing. The first step in Illumina sequencing is fragmentation of the genomic DNA and library construction. So the adapters are ligated to the ends of the DNA fragment. And then the second step is cluster generation. So these adapters, these DNA molecules are then added to a flow cell, and they are attached to the flow cell via adapter sequences, which are complementary to the short sequences on the flow cell, which are these are already present on the flow cell. Then a process occurs called bridge amplification. The next step is sequencing. So sequencing is carried out a single base at a time, and the unincorporated bases are washed away. The image of fluorescence is captured from each cluster of the flow cell. Once the laser causes excitation of the fluorescently labeled fluorophores. Then finally, we have data analysis, and these images are then transformed into base calls or reads. And in this manner, the sequence of bases can be determined. So this slide shows the bridge amplification technique for Illumina sequencing. And here, the first step is the DNA fragment to be amplified is attached to the flow cell via the adapter sequences, which are complementary to sequences that are already present on the flow cell. Then the second step is this DNA, the DNA fragment then folds over and binds to the second oligo on the flow cell to form a bridge. Then the third step is the DNA polymerase then synthesizes the reverse strand, as you can see in these images. And then the forward and reverse strand then straightens, releases and straightens, and then repeats itself to form multiple clonal copies of the forward and reverse strand in a cluster. So after this bridge amplification technique is carried out, then for each DNA fragment or read, many clusters or many copies of the read have been generated on this flow cell. Then the nucleotides and the DNA polymerase are added to the flow cell, and these nucleotides are fluorescently labeled. So they also have a terminator so that only one base is added at a time. So as you can see in these images, this process is repeated, adding one nucleotide at a time and then imaging in between. The unincorporated bases are washed away. Then the next step is the computer then detects the bases that are added to each cluster on the flow cell, and each base and each base and the cluster gives out a characteristic fluorescent signal. And this information is used to reconstruct the DNA sequences. As you can see, it gives off these fluorescent signals in these sequential images to reconstruct the DNA sequences. So coming to the ion torrent sequencing. Unlike Illumina and 454 Roche pyro sequencing, ion torrent and the ion proton torrent do not make use of optical signals. Instead, they make use of the fact that the addition of a DNTP to a DNA polymer releases a hydrogen ion, and as in other kinds of NGS, the input DNA or RNA is fragmented. And pyrosequencing is, in pyrosequencing, this is like 200 base pairs, you know. Then as in other kinds of, so then these adapters are added, and one molecule is placed on a bead, and each bead is then placed into a well of the slide. Then the molecules are then amplified on the bead by the process of emulsion PCR. So coming to this, the ion torrent sequencing, first, the steps in ion torrent sequencing include: the slide is first flooded with a single species of the DNTP along with the buffers and the polymerase, and then only one DNTP is added at a time. The pH is detected in each of these wells as each hydrogen ion is released, and it will this will cause a decrease in the pH. This pH change will allow us to understand which base was added, and also how many of these bases were added in the sequence read, to the sequence read. So coming to this, the next step here, finally the DNTPs are washed away and the process is repeated, cycling through all the 4 different types of DNTPs. So then the next step is a graph shows the pH change. Here, this graph shows the pH change with respect to the nucleotides. And the pH change, if any, is used to determine how many bases were added, and also which base was added with each cycle. So coming to the 3rd generation of sequencing. The 3rd generation of sequencing is also known as single molecule sequencing. And the 3rd generation of sequencing, also known as single molecule sequencing or long read sequencing, is a class of DNA sequencing that is currently under active development. And this 3rd generation of sequencing is considered to be one of those technologies that's capable of sequencing a single molecules, and without the requirement of DNA amplification that's shared by all the previous technologies. So there are the 3rd generation of sequencing. There are several technologies like single molecule real-time sequencing or SMRT sequencing by Pacific Biosciences, then helical sequencing by the Genetic Analysis System, and nanopore or Oxford nanopore technologies, that's MinION and PromethION, and also NGS by electron microscopy. So I will be talking about 2 of these technologies, that's single molecule real-time sequencing and nanopore by Oxford nanopore technologies in more detail in the subsequent slides. So here, single molecule real-time sequencing or SMRT sequencing. In this process, a single DNA molecule is sequenced by the DNA polymerase, and this DNA polymerase is immobilized to the bottom of ZMW. This is a ZMW. This ZMW stands for zero mode waveguide. So zero mode waveguides are named because the holes which serve as waveguides are smaller than the wavelength of light. So this light doesn't pass through it. So these fluorescent DNTPs then release a small amount of light during base incorporation. The signal is then tracked in real time, and these thousands of ZMWs, they're processed in parallel. And here is an SMRT template. The slide shows how the polymerase, which is a gray structure here, is anchored to the bottom of the ZMW. Okay, and the green portions are the hairpin adapters. These are ligated to the ends of a double-stranded DNA molecule. That is shown here in yellow and purple. These adapters that are green form a closed circle, and the gray portion, or this gray structure, is the polymerase. As I said before, it's anchored to the ZMW, and the polymerase then incorporates the bases into the read strand. That can be seen here as orange. So coming to, this is a single SMRT cell. This figure shows a single SMRT cell, and each cell contains around 1.5 lakh ZMWs. And approximately 35,000 to 75,000 of these wells produce one read per run, which lasts 0.5 to 4 hours. And finally, coming to the Oxford nanopore sequencing. So what is a nanopore? A nanopore is a nanoscale hole. An Oxford nanopore passes an ionic current through through the nanopores, and then measures the changes in the current as the biological molecules are passing through it. This information about this change in current can be used to identify that molecule. Also, these holes can be created by proteins puncturing membranes, and that is called a biological nanopores. Or if it is a solid material, they're called solid state nanopores. And you can see in this figure, the strand of DNA is passed through the nanopore, and the current is changed as the bases pass through, that's GATC, pass through the pore in different combinations. Now, here is a short video of the nanopore sequencing principle, and it would explain it in a more clear manner. So as you have seen it from the video, the tether present in the wells will guide the DNA molecule towards this nanopore and allow it to pass through it for the purpose of sequencing. And there is a change or disruption in the ionic current that will allow us to know the sequence of bases. So coming to the differences between Illumina and the third generation sequencing technologies. While comparing the second generation technologies like Illumina, that's HiSec 4000, and Oxford Nanopore and the PacBio sequel, with regard to parameters such as the read length, the number of reads, processing time, error rate, cost per run, instrument price, and advantages, the read length is highest in Oxford Nanopore of up to 900 kilobases. With Illumina of HiSec 4000, the read length is just 150 base pairs. And with the PacBio sequel, it is 10 to 15 kilobases. The processing time is less than 1 to 3.5 days with the Illumina HiSec 4000, whereas it is around approximately 6 hours with Oxford Nanopore MinION and up to 10 hours with the PacBio sequel. The error rate is less than 1% with Illumina HiSec 4000, whereas it's between 5 to 15% with the Oxford Nanopore MinION and 10 to 15% with the PacBio sequel. The cost per run is around $3,000 with the HiSec 4000, and it's between $500 to $900 with the Oxford Nanopore MinION. And the instrument price is around $900,000 with the Illumina HiSec 4000, whereas it's also that the instrument price has decreased with the third generation machines to just $1,000 for the Oxford Nanopore MinION. And the advantages of the three types of technologies are that Illumina is highly accurate, whereas PacBio and Oxford Nanopore are not as accurate. Also, both PacBio sequel and the Oxford Nanopore MinION can sequence long reads, whereas unlike Illumina, it cannot do that. And the Oxford Nanopore MinION is also a portable device. So coming to NGS data analysis overview, there are this process involves 3 steps. One is primary analysis, which involves demultiplexing, base calling, and then quality control. That's the generation of a FASTQ file. Then secondary analysis involves sequence alignment, or that's called read mapping, and the generation of a SAM or BAM file, and also variant calling, that's the generation of a BCF file. And the third is the tertiary analysis, which involves variant annotation and variant filtering. So coming to primary analysis, which involves demultiplexing, base calling, and quality control, the generation of a FASTQ file. Here, I'm coming to the FASTQ generation here. Each sequencing platform can generate data in particular formats. For instance, Illumina generates FASTQ data, and Roche generates FNA and QUAL files, whereas Nanopore generates FAST5 files. So FASTQ here stands for fast quality, and a FASTQ file generally has 4 sets of lines. The first line contains the information that's used to identify the sequence and also the sample name and other information. And this line starts with this @ symbol. The second line contains the raw reads of the sequence. And the third line contains also some sequence identifiers, as in the first line, and it starts with the + symbol. The fourth line contains quality scores for the sequences. And this quality score will tell you how accurate the read was. So coming to the Phred score. This Phred score is also known as or these quality scores are also known as Phred scores. And they're basically ASCII characters. ASCII is the abbreviated form for the American Standard Code for Information Interchange. And this Phred score is calculated using the equation Q is equal to -10 to log base 10p. And this Q score could range from 0 to 40. So coming to the FAST-QC of these sequences, this picture shows the per base sequence quality. And this is a parameter that gives you an overview of the range of quality that's measured as Phred scores across all the bases. And the score can be divided into very good quality scores. That's if these yellow bars are present in the green zone and the Phred score is more than 30. halls of reasonable quality where these yellow bars are present in the orange zone, and the Fred score is between 20 and 30, and calls of poor quality, where again these will be present in the uh, red zone, and the Fred score is less than 20. So coming to secondary analysis. Secondary analysis involves sequence alignment or read mapping and the generation of a SAM or BAM file. So here's an analogy for read mapping. Read mapping is like throwing 400 average city phone books into a paper shredder and then trying to sort out these shreds by matching them and recreating the 400 notebooks, phone books. And so it is that complicated. So, but luckily we have these algorithms and we have computer programs to do them. So coming to read mapping, mapping the reads of an experiment to a reference genome is a key step in modern genomic data analysis. With a mapping, the reads are assigned to a specific location in the genome, and insights like the expression level of genes can be gained. So for de novo assembly, or working with an organism where the reference genome is not known, then these uh, and the reads need to be assigned or assembled into contigs first. Whereas for the reference-based assembly approach, it involves mapping each read to the reference genome uh, and then identify the genetic variants that are present, like SNPs, indels, insertions, copy number variants, etc. So these are some of the tools that are used for genome assembly. Some of the main ones are Bowtie Aligner, Soap de Novo Assembler, then Velvet Assemblers, Spades Assembler, then Galaxy Tools, and Base Space Illumina. So coming to tertiary analysis, this tertiary analysis involves variant analysis, and also the resultant file format is called the VCF format, or the variant calling file format. So what is variant calling? Variant calling involves comparing the sample sequence with the reference sequence, and the sample sequence could be a single gene sequence or a whole exome or a whole genome, and comparing it to the reference sequence. And this differences between the sample and the reference can be identified. These may be single base changes such as indels or SNPs or large structural variants. And as you can see in the slide, the aligned reads are mapped to the reference genome, and a variant has been identified where thymine has been altered to cytosine. So coming to variant annotation. Variant annotation is the process of assigning information to DNA variants. And there may be different types of information that can be associated with variants. And a commonly used resource are the databases, which contain the variants that have been already described or previously described. So to find the variant IDs and the frequencies of these variants, we use databases like DBSNP, HapMap, GenomeAD, 1000 Genome. And then to find out the degree to which the mutation is pathogenic or benign, we can use databases like Polyphen, Mutation Taster, SIFT, Align GVGD. And also to find out the gene variants and its associated phenotypes, databases like OMIM, ClinVar, and LOVD can be used. And also to determine the variant in the gene and its associated disease, we can use these databases, BRCA, SHARE, BIC, and IARC TP53. So these are some of the prokaryotic annotation web servers like RAST, CRISPR Finder, MREPS, Microscope, BASIS, PGAP, and PROCA. And their associated web servers And there are also eukaryotic annotation web servers like NCBI Pipeline, Repeat Masker, Sensor, Window Masker, Segma Tool, and Maker Babylomics. Now I'm coming to the impact and applications of NGS. That's opening the doors to the world of omics. So what is genomics and the meaning of the suffix omics? You can say genomics with reference to NGS encompasses mapping the structure and organization of their genomes and differentiating between de novo sequences, resequenced genomes, exonic or targeted sequences, and metagenomic sequences. But in simpler terms, genomics is the study of all of a person's genes with and their interactions with each other and that with a person's environment. So the suffix omics could also refer to the integrated fields such as transcriptomics, metallomics, epigenomics, proteomics, and metabolomics, etc. And this is just a representative image of the central dogma of biology. So coming to the differences between genetics and genomics. So genetics is the study of heredity, whereas genomics is the study of all of the study of the entirety of an organism's genes. In genetics, a gene is a specific sequence of DNA on a single chromosome, whereas genomics refers to an organism's entire genetic makeup. And in genetics involves the study of the function and composition of a single gene, whereas genomics deals with the study of all of their genes and their interrelationships. So omics technologies, what is an omics technology? So there are various omics technologies. One is genomics, which refers to the total DNA present in a cell or organism, or transcriptomics, which refers to the total mRNA that's present in a cell. The proteomics is the set of all the proteins that are expressed in a cell or tissue, and metabolomics is the study of the global metabolite systems. So coming to this term called epigenomics. Epigenomics, so what are epigenetics, genetic processes? So mechanisms that are other than the changes that occur in the DNA sequence, and they cause differences or cause an effect in gene transcription or gene silencing. This process is known as an epigenetic process. And there are mainly two mechanisms that are involved in epigenetics. One is DNA methylation and histone modification. Also, non-coding, recently, these non-coding RNAs and small interfering RNAs have garnered a lot of attention in epigenetic processes. But coming to the goal, the goal of epigenomics, the focus of the main focus of epigenomics is to study the epigenetic processes on a large scale or a genome-wide scale to assess its effect on the disease. And coming to another term, which is metabolomics, and that is the study of the metabolome. And what is the metabolome? A metabolome can refer to the complete set of the small molecule metabolites that are present in a biological sample. And what are these small molecule metabolites? They could be metabolic intermediates, hormones, and other signaling molecules. And you can define a metabolite as any molecule that is less than 1 kilodalton in size. So coming to the metabolic phenotypes, these metabolic phenotypes are the byproducts of the interactions between the environment and the genetic processes and also the lifestyle factors. They will give you the phenotype. The interactions between them will give you a phenotype. So coming to another term called transcriptomics. Transcriptomics is the study of the transcriptomes or the entire RNA transcript that's present in a cell or cell population to investigate the gene expressions. So studying this process is performed using high throughput technologies like microarray and RNA-seq. And 95% of this mRNA does not form proteins. So therefore, by analyzing the transcriptomes, we can determine which protein is produced or not. So coming to gene expression profiling, what is gene expression profiling? Here, so to identify and characterize and the characterization of a mixture of mRNA present in a sample is called gene expression profiling. And what is the need, or what is the principle behind it? That is the amount. So the principle behind it is that the amount of a specific RNA transcripts in a biological sample is a reflection of the expression levels of the corresponding genes. And the main challenge with this transcriptome, the main challenge with a transcriptome is that in contrast to the genome, it is highly variable over time and even between cell types, and also the environment can change the transcriptome. Now coming to another branch of study, which is known as proteomics. And proteomics deals with the proteome, the study of the proteome. So proteome provides insights into the role of the proteins in a biological system. Proteomics is the large-scale study of proteins, and proteins are vital parts of living organisms they have several functions. The proteome is it's the entire set of the proteins that are produced in a single cell or an organism. And it is highly variable over time and between cell types, and it changes in response to its environment. But to some extent, this proteome reflects the underlying transcriptome also. And the protein activity is also modulated by many factors. So proteomics can provide significant biological information for many biological problems, such as which proteins interact with a particular protein of interest. And example, the tumor suppressor protein P53, and which proteins are localized to a subcellular compartment, which proteins are involved in the biological process, for example, the circadian rhythm. And the overall function of the cells can be described by the proteins and their abundance. So here is an animation of a motor protein that is kinesin, and it's dragging a vesicle, that's a storage container, along a microtubule. I was just showing you this to show as an example of what proteins do. So coming to the summary of the omics technologies. I would like to make four points in this, and that is omics technologies are primarily aimed at the universal detections of genes, that's called genomics, mRNA, that's called transcriptomics, the proteins, that's called proteomics, or metabolites, that's metabolomics, in a specific biological sample, and it has a broad range of applications. Secondly, genomics and transcriptomic research has progressed mainly due to the advances in microarray technology. Also, mass spectrometry is used for the detection of analytes and proteomic metabolomic research. And finally, data analysis is complex and a huge amount of data is generated. Therefore, the involvement of statisticians and bioinformaticians are essential in this process. So that brings me to the end of my talk, and thank you for listening.
Thank you, ma'am. Thank you for your informative presentation.
Okay.
There is a query from Anjali MP.
Okay.
Whether the metabolites are site specific, because it is mentioned as less than 1 kilodalton in size.
Yes, yeah. All so they are defined as any molecule that's less than 1 kilodalton in size. Yes, they're size specific. Hello?
Yeah, thank you. Thank you, ma'am.
Yes. Shall I
But
stop sharing the screen? Should
yeah,
I?
yes, yes, ma'am.
Okay. Okay.
Many participants are complimenting your presentation, ma'am.
Oh, thank
So,
you. Thank you so much. I can share my email. I think I see.
you can share in the chat box, ma'am.
Okay.
Yeah, I think, ma'am, that's all about the
That's
queries.
all. Okay.
Thiru sir?
Yeah. Yeah, proceed, ma'am. Yeah.
Thank you. Thank you so much.
Thank you, Maitri. Yeah.
Thank you.
Thank you, ma'am. So, on that note, we come to the successful completion of the 1 week online faculty development program. I thank all the participants for joining us and all the speakers who have presided the session. I now request Thiru sir to deliver the vote of thanks.
Blessings of our founder chancellor, we begin this faculty development program well, and we are concluding today. At the foremost, I express my sincere thanks to our honorable chancellor madam and president sir for supporting us in all means. On behalf of the Center for Ocean Research, I wish to thank our director Sashma for being backbone in all our programs and motivating us to organize all our programs. I am indebted to my guide and head of Center for Ocean Research, Dr. D. Imbakandan sir, and Dr. Stanley Abraham sir for his valuable suggestions and helping me in organizing this faculty development program. I sincerely thank to today's speaker, Ms. Maithili Verma, for her elaborate talk on next generation sequencing tools and omics approach. I also thank to all our speakers for spending the valuable time to enlighten us to expertise and making this faculty development program more interesting and informative. I am grateful to all the participants of various research institutes and college for effectively participating and making this event a grand success. I am also thankful to my colleagues of Center for Ocean Research, especially the moderator Josna ma'am and Namita ma'am for beginning this online platform and successful completion of the faculty development program. Once again, I thank all. Thank you. Thank you once again, Namita ma'am. Thank you,
Thank you.
Maitri. Thank you.
Thank you so much. Thank you.